We're going to finish chapter 11, part C, starting with 11.7, the synapse. So when we're talking about the synapse, we're talking about the junction of one neuron to another. It's going to be the immediate transfer of information from one neuron to another. So when we look at it in the blue right here, we've got our presynaptic neuron. That's going to be the first neuron, the one that is going to be communicating down the length of the axon. And then it will be communicating to the synapse, across the synapse, to the postsynaptic neuron. That's going to be neuron 2, and it's going to be communicating down the dendrites this way. So it's a one direction. So the presynaptic neuron will be conducting impulses toward that synapse. It's sending information down its axon toward essentially the synapse. And then the postsynaptic neuron is going to be getting that communication. It's going to be transmitting that signal away from the synapse. It's going to receive. So those are the two synonymous. Sending is presynaptic, receiving is postsynaptic. So when we're looking at these guys, we're going to be talking a lot about the synapse, but remember, it is a neuron. It's one direction, so it's not going to go backwards. When we're looking at the synapse, you can see how it's drawn here. This is going to be our axon coming down, and it's got all these little tendrils, and then those little tendrils are going to be tiny little gaps and then connect to the cell body and or the dendrites of the postsynaptic cell. So you can see here in an image as well. These synapses will connect a couple different ways. One way that they will connect is going to be through an axodendritic connection, which is where you will have the axons of one neuron touching or connecting to the dendrites of others. And touching is the wrong word. There is going to be a space. They don't actually directly touch. But axodendritic is an axon to a dendrite, whereas an axosomatic is going to be an axon to the cell body. You rarely are going to have axon to axon, dendrite to dendrite, or the dendrite to the soma. So basically, the axon's connecting to either the dendrite or the cell body of that postsynaptic cell. And there's two types of synapses, a chemical or an electrical synapse. So the most common is a chemical synapse. We've already talked about this in reference to the neuromuscular junction. Essentially, that's what we're getting ready to talk about again. So chemical synapse is the most common. It's going to be releasing a chemical. We call that a neurotransmitter. And that neurotransmitter is going to be released across this synapse. And so there's going to be two parts of this synapse. You're going to have the synaptic vesicles that are leaving the axon terminal in the presynaptic neuron and is going to then travel, and that synaptic vessel is going to release that neurotransmitter to the postsynaptic neuron, which will receive the transmitter. And that space between the two is going to be called the synaptic cleft. So basically, you have an action potential occurring, which is electrical, that changes to a chemical synapse, and then that changes back to an electrical. So here we've got our presynaptic cell. We're sending a signal down. This is electrical. We're going to have these neurotransmitters travel across that synaptic cleft. This is chemical. And then once we receive them on the other side, we're going to cause graded potentials, and it's going to be back to an electrical signal. So it kind of goes back and forth, which is very cool, in my opinion, that it does that. Okay, so now when we're looking at these chemical synapses, there's an image in the PowerPoints if you guys would like to go look at that. But with a chemical synapse, there are 6 steps in order to transfer the information across a synapse. I'm going to just go through these as an overview because, again, these are the exact same thing we've pretty much already talked about in relation to the neuromuscular junction. So we're just going to kind of go through it high level. 6 steps. The first one is you have an action potential at the axon terminal. So that means if this is my lovely little neuron, I'm sending here's my dendrites, here's my axon. We're sending the signal down this axon. It's going to arrive at the very end at that synapse, at the terminal of the presynaptic neuron. Then we're going to have calcium. That calcium is going to open, voltage gated calcium channels are going to open in your presynaptic axon terminal. And sodium is going to go in. So our sodium is going to enter the axon terminal. When that sodium not sodium, when our calcium, he's going to be coming into the cell right here. When our calcium comes into the cell, he's essentially going to release these vesicles that are going to be holding this neurotransmitter, and it's going to dump into your synaptic cleft. So, like I said, our calcium is going to cause the synaptic vesicles to release that transmitter across that synaptic cleft. That is going to occur because of something called synaptogamenin, which is a protein that causes the synaptic vesicle to find that plasma membrane, join with it, and then essentially like pop open and kick everything out. So that is happening for step 3. So now we have our neurotransmitter floating across this synaptic cleft. So as this neurotransmitter is diffusing across the synaptic cleft, it will then bind to receptors on the second synaptic membrane. So basically the neurotransmitter has to bind to the correct thing. So the key and the lock have to fit together. When the neurotransmitter binds, it will open that channel. And if it opens a channel, as we know, ions will move. And when ions move, we get graded potentials, not an action potential, a graded potential. It's local. That graded potential is going to cause that excitatory or inhibitory event. Either the resting membrane potential will become more positive or less positive. That's basically what that means. These are all images to kind of relate to all that. Part number 6 is where the neuron essentially will terminate the effects of that neurotransmitter. If it stays in this space for very long, it will keep binding to the receptor on the postsynaptic neuron, and it will keep opening. You'll keep having the effects. So basically, we need to make sure we go through and clean the slate and wipe out anything in the synapse, so that way the neurotransmitter doesn't continue doing what it's doing. So there's a couple different ways it can do that. Some of these terms are going to be reuptake. Reuptake basically means that the axon terminal will like suck it back up. Degradation, it will be destroyed by an enzyme in the synaptic cleft. Or diffusion, it will essentially diffuse away from that location. So those are the 3 ways that your neurotransmitter effect can be stopped. So you've got an action potential as the first step. You open voltage-gated calcium channels. Calcium enters the presynaptic terminal. Then you're going to have your synaptic vesicles are going to fuse with the synapse, and are going to open up that neurotransmitter into that cleft. It's going to diffuse across that space, bind to receptors on the postsynaptic neuron, and then at some point it's going to do whatever it needs to do, cause a graded potential, and then we have to get rid of the neurotransmitter, and we have to somehow have it degrade. So that's what's going on with this synapse. Communication from one cell to another. So with a chemical synapse, the one we just talked about, we have all of those 6 steps happening, which means it's slow. There is a delay for a chemical synapse, so much so that it can be 0.3 to 0.5 milliseconds of a delay. That does not seem like a very long time, but that actually is a decent amount of time that there's a delay in some sort of communication. We want communication in our neurons to be very, very fast. The second type of synapse, electrical synapse. This is a little bit less common. It's not going to be found everywhere. It's specifically going to be found in your heart because it's going to be formed with gap junctions. So that's one area you may see it. You also see it in some brain regions. But an electrical synapse is the neurons are actually joined by a gap junction, which is this lovely little guy, which means that there's a tunnel between the two. So the plasma and the all of the fluid is connected between the two. So an ion is moving through the fluid. It's not having to cross a plasma membrane. So it's a little bit quicker. It's a little bit more fluid, and it can be bidirectional. So communication can go both ways. Okay, so we have said that a postsynaptic potential, we've talked about action potentials, we've talked about graded potentials, and this is a postsynaptic potential, not to get them all confused. A postsynaptic potential is a graded potential. So remember, these are short distances and they're not very, they don't last for very long. So a neurotransmitter, when it is actually communicating down the axon to another cell, and you've got your postsynaptic cell that is taking up that neurotransmitter, a postsynaptic potential is whatever is occurring in the postsynaptic cell. Whatever is happening in the second neuron is called a postsynaptic potential. There are two types. You can have an EPSP, excitatory postsynaptic potential, or you can have an IPSP, inhibitory postsynaptic potential. So basically, when communication crosses that synapse, that cell, the postsynaptic cell, will either get excited and have an EPSP, or it'll be inhibited and have an IPSP. So we're going to break down those two. An EPSP is very similar to an action potential. It's not, but it's very similar. It's an excitatory postsynaptic potential. And what is happening is you're going to open chemically gated channels. The ones we like to open are the same ones in an action potential. Sodium will open, and when sodium opens, you'll have sodium flow into the cell. You have more sodium opening and more sodium flowing in, so you have a change in the more positive direction. You are going up the graph right here. You do also have potassium open, and you do have potassium flowing out. So remember, potassium and sodium are flowing in opposite directions, but it's both a positive ion. But in this case, in an EPSP case, the sodium influx is greater than the potassium. So there's more sodium moving. Therefore, you have more depolarization. Therefore, an EPSP is a change in the graph going up. It's excited, more positive or less negative, EPSP. Or you can have an IPSP, which is the exact opposite, inhibitory postsynaptic potential. It's going to be hyperpolarization. With an IPSP, you are opening potassium, but you're also opening chloride channels. You are not opening sodium. An IPSP is opening potassium and chloride. It is not opening sodium. So you're going to have your positive ion move out. So you're taking all of that positive ion with it. There's not enough chloride movement. It is moving in, but there's not enough movement in to counteract that potassium moving out. So therefore, the membrane will become more negative and it will go down, so that deflection in the graph. That's an IPSP. So with this EPSP and this IPSP, either way, you're getting some sort of small signal happening right at the location where the neurotransmitter binds. But what you can do is add up these effects, and that is called summation. So summation is where you are adding up. Think of it like a math problem. You can add EPSP and EPSP and get two positives. So you've essentially added two things. You can add an EPSP and an IPSP and you've added a positive and a negative. You can add two IPSPs to get more negatives. So you can kind of summate these effects. So there's two types. You can have temporal summation or spatial summation. Temporal summation is basically just saying that the effects are going to be added from one or more presynaptic neuron at the same time. So this is basically saying that there are two people and they both say whatever they need to say at the exact same time. If two people are talking at the same time, it's loud. Temporal summation. One voice isn't bad, but two voices talking at the exact same time is louder. It's a stronger signal. Spatial summation. You're adding the postsynaptic from multiple inputs. The spacing of where the graded potential is happening is added together. So that means if I am standing in one spot, someone else is standing 10 feet away, someone else is standing 10 feet away, and there's another person standing 10 feet away, If we all then say something back to back, we all add together, that's going to be a stronger communication as well. So spatial summation is over greater places, multiple inputs. Temporal summation is at the exact same time. Okay, so that's how we can essentially add. So if you look right here for our postsynaptic, I'm trying to make this bigger, but I'm going to end up not making it work effectively. So here we've got an EPSP, upward deflection. We can add those two together. So this is going to be no summation essentially. It's just two separate communications. But we can add them together. So this is temporal summation. So this is going to be really close together at the same time. You can see that happening. So here's one, here's the other. If we add those together, we essentially make it a large deflection. Or we can add it because it's hitting this place and this place at the exact same time, but in different locations, we're going to add them together. But these are both EPSPs, so they're both excitatory, so they're both going up on the graph. You can also add IPSPs. So this one is going to be an EPSP plus an IPSP. Basically, we add an upward deflection, but then we have a downward deflection, and it takes the graph essentially back to baseline. So you can kind of see how all of that works together. Okay, so there's another term that we're going to talk about before we move on to neurotransmitters, synaptic potentiation. Synaptic potentiation is going to be where we have repeated use of the synapse. And if we have repeated use of the synapse, it's going to increase the likelihood or the ability of that cell to excite the postsynaptic neuron. Basically, the more you use it, the more it'll fire. Potentiation, it will add. So repeated use of a synapse will increase the likelihood that it's going to actually get excited in the long run. Potentiation is a big area of research, specifically when it comes to learning and memory. That's kind of where they're thinking some memories come from, because if you are using it a lot or learning, being able to remember things, repetition, if you've ever heard that, repetition is the key to memorization, the key to learning. You essentially do multiple things multiple times, you will remember it better. They think that's related to the synapse. Okay, there's also something called presynaptic inhibition. And basically what that means is that there can be both an EPSP and an IPSP happening from two different neurons that are presynaptic onto one postsynaptic cell. So you can kind of see how we have one pre, a second pre, and then we've got our blue postsynaptic cell. Therefore, it will have whatever effect it's going to have, it will have a smaller EPSP. So that's going to be inhibition, where there's a different neuron altogether giving it communication. Okay, we're not focusing on the endomembrane system for this chapter, so you can just ignore all of these slides. And then this last section of the chapter that we're going to talk about is going to be 11.9, neurotransmitters. So neurotransmitters is basically the communication, it's the language. If our neuron is sending a neurotransmitter from one neuron to the other, it's the language. Whatever the neurotransmitter is what is communicated. There's about 50 or more neurotransmitters that have been identified. Most neurons have two to a couple different kinds, but it does seem to be that neurons are more specific. They don't have all 50 of them, but they can exert a couple different kinds of communication because they may have more than one type of neurotransmitter that they can release. So a neurotransmitter is going to be classified by two things: the chemical structure that makes it up, and then what the function is. For the chemical structure, we're not going to go dive deep into that because this is not a biochem class. We're going to focus more on the function. But real quick, to cover chemical, but we're not going to dive in too deep. Chemical classification is going to be based on whatever the molecular structure of that neurotransmitter is. It's very biochem based. It's very 3D. It's very atom generated. It's not necessarily my wheelhouse, even though I really did enjoy it in biochem in grad school. We're not going to spend a lot of time here. So just some terms you need to know. One type of neurotransmitter is acetylcholine. We've talked about it in regards to the muscular system, ACH. It's the first one that was identified. It's the best understood. It's released at that neuromuscular junction. We talked about that in chapter 9. We will also talk about this again in chapter 14 with the autonomic nervous system. And we see it in some central nervous system neurons. So this guy is pretty generic and covers a lot of things. And he is synthesized from two different molecules, acetic acid and choline. And he is broken down in the synapse by acetylcholine esterase, that enzyme. But we also alluded and talked about that in chapter 9. Okay, then we've got another structure called biogenic amines. Amines are basically going to say that there's going to be a nitrogen somewhere in the structure. Again, we're not going to dive too deep into that. You don't really need to know the difference between the catecholamine and the endolime, but a biogenic amine is going to cover dopamine, norepinephrine, epinephrine, serotonin, and histamine. Whew. So let's talk through those. Dopamine is going to be a neurotransmitter that is related to mental health, schizophrenia. They have found that a schizophrenic diagnosis is usually going to be paired with an increased level of dopamine. So there's some communication and they think is going on in that world. Norepinephrine and epinephrine are also called adrenaline and noradrenaline. So epinephrine and adrenaline are the same thing. This is the neurotransmitter that is occurring when you get in a car accident, but you don't feel any pain because you've got that adrenaline running through your system. That's the epinephrine. Whereas you're coming down from it, the norepinephrine is going to be the opposite. So those are those two. And then the serotonin and histamine. Histamine is going to be usually related to an immune response. So during this lovely season of pollen that we've got going on right now, a lot of people are taking antihistamines, and basically it is going to be a medicine that is targeting your histamine receptors and saying, you don't really need to work right now because histamine is going to cause inflammation in your system. It is increasing when you need to fight off some sort of something, i.e. allergies. Then we've got serotonin. Serotonin is also related to mental health, usually going to be involved in depression type diagnoses. So if there's a depression, usually it's going to come with a decreased level of serotonin. So one of the common medicines to treat depression is called an SSRI. And an SSRI is selective serotonin reuptake inhibitor. So basically, selective serotonin, some serotonin, reuptake inhibitor. It stops the reuptake by that presynaptic cell, so it stays in the synapse longer. If it stays in the synapse longer, we get more serotonin binding to our postsynaptic cell, which means we get more effects of serotonin. Smaller amounts of serotonin, we usually have a diagnosis of depression. So that's kind of how those all work together. It's very cool. A couple other things that are considered neurotransmitters, amino acids. So glutamate, aspartate, glycine, GABA, some of the things we talked about, building blocks of proteins, can also act as an amino acid in the nervous system, which is very cool. A couple other things, endorphins are a neurotransmitter. ATP can act as a neurotransmitter. And then there's some gases that can act as a neurotransmitter. Nitrous oxide and carbon monoxide are two of the big gases that we know can actually be a neurotransmitter. So this is not their sole function, but they can also act as a neurotransmitter. So another one that we actually have is we do have a neurotransmitter that is an endocannabinoid that will actually act on a cannabinoid receptor, which that cannabinoid receptor is natural to us, and it is the same receptor that cannabis from the marijuana plant can bind to. So you do have a receptor that is built for the chemical structure of cannabis. So that's just an interesting kind of biological mechanism that we have. Okay, so these are all of the chemical structures. We're going to spend a little bit more time on the classification of this chemical structure and then the function. So When we're talking about the function of a receptor, let me find it. There are different types of functions it can be. There are two main ones. It's going to be an effect or an action. This is how we we'll break down the functional classification of a neurotransmitter. The effect, a neurotransmitter will either be excitatory or inhibitory. Excitatory means that when it binds to the postsynaptic cell, it will cause depolarization, the EPSP. If it's an inhibitory effect, it will cause a hyperpolarization or an IPSP. So that's the effect. It's whatever the depolarization or hyperpolarization that occurs in the postsynaptic cell. Some neurotransmitters are going to be one or the other. Some neurotransmitters have both effects. So for example, acetylcholinesterase, ACH. We know that it is excitatory. It causes depolarization in the neuromuscular junction. It turns on a muscle contraction. But if you are ever having a heart attack, you do not want ACH because it will inhibit cardiac muscle. So you don't want to stop your cardiac muscle from moving. So you don't want to inhibit it. So depending on where it's used, acetylcholine actually can both excite or inhibit. That's very interesting. Whereas the action will be how it has the effect, where it affects. There's a direct and an indirect action. The direct action is basically the neurotransmitter is opening and closing the ion channel directly at that postsynaptic cell, postsynaptic membrane. It's all happening right there. Everything we've talked about so far in this chapter is direct. Indirect, we have not talked about yet. Indirect is basically where the neurotransmitter is going to bind. It is not causing either an EPSP or an IPSP. It's causing something else communication to happen. It's called a second messenger system. They can have upwards of 6 steps. They're very, very long. We're not going to spend a lot of time on them, but it usually involves something called a G protein coupled receptor. So there should be an image way down here. Let me drive down to it of this second messenger system. So this G protein indirect action is basically the neurotransmitter binds here. Rather than causing the graded potential right here, it causes a step here, a step here, a step here, ooh, another step here, a step here. This guy has to do something, and then this. So an indirect action is basically triggering a cascade effect so that other things are occurring in order to eventually get to some sort of reaction of whatever it needs to be. So everything we've talked about is basically direct except for those indirect cases that you're going to hear about coming up in class. A lot of biological processes are going to be an indirect communication pathway. And unfortunately, you are going to need to know all of these G protein coupled receptor second messenger systems. You don't need to know it for my class, hallelujah, but when you get to nursing school, physiology classes, any kind of your next level of academics that you're going to do, more than likely you're going to come across these and you're going to need to memorize quite a few. But for my class, all you need to know is that they exist and it's an indirect path. We are not focusing on 11.10. We're not going to look at neural integration and processing, so that is where we're going to end part 3.